In this lecture, we'll be focusing on catalysis, specifically enzyme catalysis, since we're looking at proteins. So what does a catalyst do? Well, a catalyst essentially speeds up the reaction without being consumed, and it does so by lowering the activation energy. So if we look at an energy landscape with a reactant to the product, in the absence of an enzyme, you have to overcome this activation energy in order to get to product. And so what a catalyst would do, or an enzyme catalyst would do, it would reduce this activation energy, right, in the presence of the enzyme so that we can get to the product. And essentially the activation energy is the amount of energy required to reach this transition state. On the blackboard in the classroom, I'll be going through mechanistically catalysis, specifically in enzymes. And so this is just to remind me to tell you to look at your notes. So nucleophilic catalysis involves a nucleophile that allows for the catalysis to occur. And so there are several types of nucleophiles. Shown here in this table are a series of different residues that can serve as nucleophiles, including serine, threonine, cysteine, aspartic acid, lysine, histidine, and tyrosine. And each of these nucleophiles have a corresponding enzyme, and then these are the intermediates for all of these enzymes that you can go through. So instead of using water as a nucleophile, the enzyme produces, in this case, serine hydroxide to do the reaction because alcohols are better nucleophiles. Serine reaction is intramolecular and favored entropically, and the arrangement of the group is more rigid and defined in the catalytic area, allowing for the process to happen very rapidly. The principle of macroscopic reversibility, or detailed balance, is used in thermodynamics to place limits on the nature of transitions between different quantum or other states, and applies to chemical or enzymatic reactions, as we're talking in this lecture. Transitions between any two states take place with equal frequency in either direction. So essentially, the principle states that the reaction pathway for the reverse reaction at equilibrium is the exact opposite of the pathway for the forward. So the transition state is identical for both the forward and the reverse reactions. So here's an example that summarizes the classic factors that we've discussed on enzyme catalysis. So here is the enzyme, and here's the substrate. So here's the enzyme substrate complex, in which you have the histidine serving as a general base, deprotonating the nucleophile, serine in this case, which then makes this nucleophilic to do the reaction and react to this electrophilic carbon. And this is stabilized by residues electrostatically in the pocket. Now, once this is done serving as a general base, it becomes a general acid, right, because it's now protonated. And now rearrangement is happening from the transition state where your oxygen electrons is coming down. This bond is being broken, right, so you're taking an amide bond and you're hydrolyzing it. And as a result, you have this enzyme stabilized again with histidine and serine. Here's your amine that has been released, and here's your serine nucleophile is still attached to the intermediate. And so once you have this, you have electrons from your nitrogen again taking water this time. This will make water nucleophilic. The nucleophile will react with the serine that's conjugated to this substrate, and this will then form this transition where now once the water has attacked, now electrons will come down and and release so that you now form the acid, and then you have your full product. So from the things that we discussed on the blackboard in terms of the different types of catalysts and the different classifications, you should be able to, on your own, take something like this and identify which one's the general base, which is the general acid, etc. So in biochemical reactions, reactants are commonly known as substrates. The catalytic events that convert substrates to product involves the formation of a transition state, as was discussed before, and occurs most easily at a specific binding site on the enzyme. That site is called the catalytic site and has been structured evolutionarily to provide specific high affinity binding of substrates and to provide an environment that favors that catalytic event. The complex that forms when the substrates and enzymes are combined are called the enzyme substrate complex, ES. And the reaction products arise when the enzyme substrate complex breaks down, releasing the free enzyme. So at minimum, an enzyme substrate complex must be formed, must pass through a transition state, which is represented by enzyme substrate with a star, and the transition state complex must advance to an enzyme product complex, or EP. And then once this product complex is generated, it then dissociates into the enzyme again and product. And so essentially you take enzyme plus substrate, it forms this enzyme substrate complex, which then passes through this transition state, this enzyme substrate transition states, which form the product and then releases the product. The enzyme kinetics gets complicated very quickly. So if we consider this unimolecular reaction where you have enzyme plus substrate forming that enzyme substrate complex, which forms the enzyme product, and then it releases that. You can look at the rate of product formation as the rate, right, K3 of the enzyme product complex minus K minus 3 which of the enzyme and product. And then if you substitute the enzyme product as a function of K2, K minus 2, K3, and K minus 3, the enzyme is the enzyme substrate complex. The enzyme product from enzyme and substrate is enzyme substrate from substrate, initial substrate, etc. So then this whole equation gets complicated. So it's important that we simplify these things in order to at least start to understand about the kinetics of enzyme. The Michaelis-Menton equation simplifies kinetics, where now you have enzyme plus substrate, which then leads to an enzyme substrate complex, and then that leads to enzyme product formation. And so you have K1 and K minus 1 in the case of the enzyme substrate to the enzyme substrate complex, and then K2 forming the product. Here, Michaelis-Menton also uses the convention of this being the Ks and this being the kcat. And so in this, the velocity is defined as the rate of product formation, which is then equal to K2 times the enzyme substrate concentration, right, as shown here. And then the overall rate of enzyme substrate complex becomes equal to K1 times enzyme times substrate minus K minus 1 enzyme substrate and minus K2 enzyme substrate. Also, when looking at the velocity, when you substitute instead of K2, kcat, velocity is then equal to kcat times, enzyme substrate. So please bear with my handwriting here. Two major assumptions are made in terms of Michaelis-Menton. One is that there's an assumption of equilibrium, and two is that there's an assumption that it's undergoing steady state. And so in the assumption that it's under equilibrium, your Ks, right, which is the K minus 1 over K1, is then equal to the enzyme times substrate divided by the enzyme substrate. And then in terms of assuming a steady state, this means that the formation of the enzyme substrate complex is then equal 0. And also an important thing to note is that the enzyme in this case, or the concentration of the enzyme, is equal to the concentration of the enzyme minus the enzyme substrate. As in the previous case, the rate of product formation was defined as kcat times the enzyme substrate complex. This can be converted into an equation where it's now in terms of the total enzyme concentration as well as the substrate, as shown here, where now the rate is equal to kcat times the total enzyme times the substrate divided by km plus the substrate concentration. So here, again, this is your rate of product formation. Kcat is your turnover, and it's always first order. That the enzyme substrate complex decomposes into the enzyme product. Km, shown here, is the Michaelis constant. It's always an apparent enzyme substrate dissociation constant, and then E naught is your total enzyme. So now I've made more room because there is a version of the Michaelis-Menton or the Briggs-Haldane version in which now the velocity is equal to the enzyme and the substrate times the substrate. So this is the total enzyme, E naught, times the substrate times K2 divided by the substrate, and that is plus the K2 plus K minus 1 divided by K1. And so in that case, if we look at the Briggs-Haldane, kcat is equal to K2, Km is equal to this K minus 1, K minus 1, K plus 2, shown here, divided by K1. And there are more complicated mechanisms, more complicated Kms, other than Briggs-Haldane. So Michaelis-Menton kinetics can be determined by plotting a Michaelis-Menton curve, and shown here, you plot the velocity as a function of the substrate concentration. And when you do plot the initial velocity as a substrate concentration, you're going to get a plot of which the asymptote becomes the Vmax. Half of Vmax will equal to Km, and so you will then be able to determine all the important kinetic values from this plot. kcat, the catalytic constant or turnover number as defined by the Vmax over the total enzyme concentration, is the inverse of time taken for 1 mole to complete reaction sequence, and the units are reciprocal seconds. For n amount of steps, the time for the catalysis is equal to the sum. Same thing for 1 over kcat is equal to the sum of the different rates. And the kcat is less than or equal to the minimum K1 all the way to Kn. Km, the Michaelis constant, or also known as the equilibrium constant, like I'd shown you before in the Briggs-Haldane mechanism, Km is equal to this. There's special cases in which K minus 1 is much greater than K2, of which then the Km will equal to K minus 1 over K1, which is essentially the real dissociation constant. This case, where you have the k minus 1 much greater than k2, is implied in the Michaelis-Menton mechanism, and it's an additional assumption for the Briggs-Haldane equation as shown here. Generally, the Km is equal to the enzyme concentration, substrate concentration divided by the total enzyme substrate. This total enzyme substrate means all the enzyme substrate and all the enzyme product species. And values of Km range from about 10 micromolar to 100 or 10 micromolar to 0.1 molar. And this depends on the enzyme, substrate, pH, and temperature. As I had said, as I had shown you before, in the case of a Michaelis-Menton plot, your Km is half the Vmax. So as you note, here's the equation of your rates. Velocity, let Km is equal to the substrate concentration, and then if that's the case, the V is going to be half Vmax, and that's how you're able to determine Km. So, so when you run experiments, if you find the asymptote and find the Vmax, then you can identify half the Vmax to identify the Km. So Michaelis-Menton equation is used for many mechanisms. They're a good fit to experimental data. You don't get enough data to get all the different Ks or rates. And so you're okay just identifying the apparent dissociation constant, Km, and the turnover, kcat. Vmax, sometimes you don't know the actual enzyme concentration. You don't know the molecular weight or how much of it is active. So since Vmax is equal to the kcat times the enzyme concentration, it's not an intrinsic property, it's actually dependent on the concentration. You can then get away with, as you might read in some of the papers, with just looking at the Vmax values. The maximum rate when the enzyme is saturated with excess amount of substrate is shown here. And so then the velocity just becomes equal to the Vmax. Some important features about the specificity constant, or kcat over Km. It's the apparent second order rate constant. It relates to the properties of the free enzyme and the free substrate. Relates the reaction rate to the concentration of the free rather than the total enzyme. At low substrate concentrations, the enzyme is largely unbound, so E is approximate to E naught. As a result, your velocity is now just kcat over Km times enzyme times substrate. Just the specificity constant determines the specificity for competing substrate, and it sets a lower limit on rate constant for association of enzyme and substrate. Basics of Michaelis-Menton experiment. What you want to do is you want to plot the initial rate versus substrate. And the reason why we do so is because the mechanism assumes product formation is irreversible, right? K minus 2 is equal to 0. There's no such thing as K minus 2 in the Michaelis-Menton mechanism. So if K minus 2 is not equal to 0, you measure when there's no product concentration, so there's no back reaction. And this was the assumption used to derive that Michaelis-Menton equation. And then you want to avoid enzyme degradation as well as inhibition, because that can then also impact how the rates are evaluated. So using this equation, you then measure the initial velocity as a function of substrate concentration. And then you are able to then identify the Vmax. Again, half the Vmax to determine Km. So we're looking at product formation. As a product is being formed, if we plotted just and looked at the substrate, and the substrate will be declining. When we look at this region over here, can't measure the time at 0, and it's not an average rate between minutes 1 and 3, right? But it's extrapolated to figure out what time 0 is. So when we look at data processing, like I said, for Michaelis-Menton, you plot initial velocity as a function of substrate concentration, but it's not an accurate way to determine the kinetic parameters because you have to wait till and see if there is a plateau that's occurring, and depending on how you're accurately measuring this Vmax, then you can obtain all the other kinetic constants. The other way to get a little bit more accuracy in terms of identifying the Vmax and the Km is to perform a Lineweaver-Burk plot, in which you plot the reciprocal of velocity over reciprocal, 1 over s substrate. And in this case, when you plot that, the point over here, you're going to hit 1 over substrate. This would be 1 over Km, right? So you can identify Km, and then when it hits the y-axis, you'll get Vmax, and the slope is equal to the Km over Vmax. So here is just a summary of the different mechanisms of Michaelis-Menton kinetics and how complex it can get. I had mentioned earlier about the Briggs-Haldane version, which I've shown, but you can actually get enormously complex. For this case, at least for our course, understanding general Michaelis-Menton as well as the Briggs-Haldane version should be sufficient to at least get you through the papers that we'll be discussing. The other thing that we need to look at in terms of kinetics, enzyme kinetics, is inhibition. So there are different types of inhibition. One shown here is called competitive inhibition, in which the inhibitor actually competes against the substrate to form an enzyme inhibitor complex, which leads to no reaction, rather than going through the productive route and forming the product and the enzyme. In competitive inhibition, your possible mechanism is that it actually binds in the active site, competing with the actual substrate. So instead of having your enzyme substrate forming your product, you have this competing process in which you have a Ki that is due to the formation of this enzyme inhibitor dead-end complex. So you can have a Km that's determined, etc., but it won't be the actual true Km because of this inhibition occurring. The kinetics in the presence of inhibitors looks like Michaelis-Menton with modified constants. So with competitive inhibition, instead of having this equation shown here, you're now having the addition of this inhibitor concentration, and this can be shown as an effect in a double reciprocal. So here's an example of competitive inhibition. Shown here in green is the normal kinetics, and what happens in competitive inhibition is that the Vmax is unchanged, yet the Km is changed. So you have an increase in Km occurring in an competitive inhibition prop. So in addition to competitive, there is non-competitive inhibition. Again, this is the enzyme without any inhibition. And here is what happens when you have a non-competitive inhibitor. What happens is you have your Km is not changed. However, you're lowering your Vmax, as shown here. So it's affecting the rate without affecting the substrate binding. So some possible mechanisms for this is that there's a very small addition to the active site, like a proton or cation. You have removing of inhibitor binding that affects the exact positioning of catalytic groups. Then you can also have a mixed non-competitive type of inhibition where you have, right, this is your inhibitor that is competitive, and then there is your inhibitor that is non-competitive, which forms this enzyme substrate and inhibitor intermediate, which is not productive. And in this case, when you have mixed inhibition, here's again your regular enzyme, but then in mixed inhibition, now you're going to have both the V and Vmax and the Km being changed. So in this case, you're lowering the Vmax and then you're raising the Km. So in an uncompetitive, just looking at that, because in the previous case we had a mixed competitive, you have your enzyme and your inhibitor forming this enzyme substrate inhibitor sort of complex, which then is not productive. In uncompetitive inhibition, you're looking at, here's your enzyme without any inhibition. Uncompetitive inhibition, you're also changing both the Km and Vmax, and they're changed by the same exact factor, as you can see. You're seeing a loss in Vmax and a loss in Km. So the possible mechanism for this is that the inhibitor binds to the enzyme substrate complex, not at all to the free enzyme. And this is not very common, but there's examples where inhibition of myo-inositol monophosphate by lithium serves as an uncompetitive inhibitor. The experimental product of inhibition is uncompetitive. So this is a summary of all the types of inhibition. We talked about competitive, non-competitive, mixed, and uncompetitive. And it's my expectation that you should be able to identify those. Reading the papers, you should be able to identify those, but also in your problem sets, being given values and then determining by plotting these whether a particular inhibitor is undergoing any of these.